Welcome, Life Sciences. Welcome to another exciting life science lesson. We are going to recap the cell. We're going to go through all of the important parts, just hopefully all those terminology comes flying back, all those diagrams coming back to you. It's really important at the end of each section that you do go back. I'm hoping that you maybe have got summarized notes, that you have got summaries, that you've done little notes on the section, because obviously it's a really important section that you need to be able to do. It's one of the foundations for the rest of the sections that we're going to carry on with. Okay guys, so where did we start off? We looked at this whole concept that if we're going to look at cells, all right, we need to be able to see cells. Let me get my pen going over here. So how do we see cells? So if we look at technology over time, technology, all right, is able us to see the smallest things, and that is the microscope work. As I said, I hope that some of you do have access to microscopes, right, at your classroom. If not, it's unfortunate, but the whole concept of a microscope is that I can see something that's really small, I can see it bigger. And this is what has enabled us to see cells, right? To be able to see what they look like, to be able to tell us what they do, is this whole concept of technology. So throughout time, what have we been able to see? Microscopes have got better and better and better. Remember, you had the light microscope maybe at your school, then at really big or universities and laboratories, they've got transmission, all right, electron microscopes, and all right, your electron transmitting microscope, okay, your LEM, your TEM, and your SEM. So we've got, with this whole thing, we now have a better idea of what cells look like. Now, when we look at this concept of a cell, there's just two terms that we need to look at. A cell can be either a prokaryotic cell or a eukaryotic cell. And what do I mean by that is quite simply, if we look at a timeline, right, of throughout time, prokaryotic cells were one of the earliest living cells. They are unicellular, all right, unicellular. They are only one cell, yet they are able to, all right, do the whole, as I said, Mrs. Greef thing. They are able to undergo all the living characteristics that we looked at. They are quite primitive, right, in their being. They are oldest today, right? They are bacteria. They have the whole kingdom called, all right, the kingdom Monera, and they are prokaryotes. And simply what they are is they have a cell membrane, they have cytoplasm, but they do not have a nucleus, and that was very important, and they do not have, all right, any membrane-bound organelles. So they had a cell membrane, and they had cytoplasm, and they usually had some kind of DNA, and it was not in a membrane. Then as time went by, all right, organisms became more complex, and with that complexity, needed a cell that could do, all right, far more complex things. And hence that cell is the eukaryotic cell. Now, when we look at the eukaryotic cell, we are going to divide that cell into a plant cell and an animal cell. And remember, guys, you must be able to tell the difference between a plant cell and an animal cell. Now, when we looked at this concept of a cell, we noticed that all living cells, especially eukaryotic cells, have three basic things in place. Number one, they're all surrounded by a cell membrane, okay? And that cell membrane kept everything in and kept everything out. So you have this enclosure. Then we looked at this concept of cytoplasm, and as I say to you, you need to think of a jar of jelly, making a bowl of jelly. That's what cytoplasm was about. And then inside the cytoplasm, you are going to find structures, little structures that have got certain functions. Now, all the cells that we're going to look at, all of them have a nucleus. And we're going to have a look just now what that nucleus is. Simply, it means a structure by itself. It's separate to the cytoplasm, right? It's in the cytoplasm, but it has a membrane of its own. And most importantly, we're going to have a look just now. It's got that chromatin network where we carry our hereditary characteristics. So we started off by looking at what the cytoplasm is. And the cytoplasm, remember I said to you, is that jelly, right? So we take, there we go, the jelly, we take water, we take the jelly, we put it together, and that's what we're going to get. So in here, the most important constituent is water, right? Water makes up approximately 60% of all the cells. Then dissolved in the water, we're going to find gases and all our mineral salts. Now, also in the cytoplasm, not necessarily, right, dissolved, we're going to find our proteins, our carbohydrates, and our fats. Now, guys, remember the whole thing about is we eat food. We eat food because we need it for energy, and we need it to build our bodies. So when I ask you about this concept of build, what does building mean? Building simply means we need to build cells. So the food that we eat, all right, goes towards making these cells work. We need to build them properly. If we build them properly, they are going to work properly. Now, when we looked at this concept again of a cell membrane, cells have got these boundaries. What is a boundary? It means the outside. And the reason why I've drawn a plant and an animal cell, or I've given a picture of a plant and animal cell here, is because plant and animal cells will have different boundaries. Now, if we have a look here, an animal cell has one boundary, and that is going to be the cell membrane. If I look on this side over here, I'm going to see that the plant cell also has a cell membrane. So all living cells are going to have the cell membrane. But the plant cell has an extra boundary on the side, and that is going to be the cell wall. Plants need a little bit of extra support. Plants need, all right, to keep their shape, and so they're going to need a cell wall. So if we have a look here at the structure of the plant cell, right, what is a cell wall? We only find it in a plant cell, very important. It allows everything in, and what's the most important component is cellulose, right? And if you can remember in your biochem, cellulose was a polysaccharide, right? So it's made up of, it's a carbohydrate, and it's made up of a saccharide, the polysaccharide, like starch and everything. And here we have the cell wall, nice and strong, non-living, to be able to support the cell. Plants, the cell wall also needs to have spaces in which they can communicate, and they do so via these little holes or these passageways called the plasmodesmata. Now, when it comes to the cell membrane is quite a complicated, all right, structure. And we give the name a fluid mosaic model. And what do we mean by that is the fluid means that the cell membrane can move. When we go to our plant cell, the cell wall over here, we notice it's very rigid. And another word I would use there is permeable. What do I mean by that? Is the cell wall is going to let everything through. However, when it comes to our cell membrane, we can say that our cell membrane is living, it's alive, right? It's a fluid molecule. It needs to be that because very often animal cells need to be able to move. What do we mean by a mosaic? there are structures, there are carbohydrates, there are proteins that fit into this fluid mosaic molecule, and these little phospholipids, the bilayer, and this whole structure of the cell membrane, right, is to facilitate, we're going to look at that just now, right, transport. The most important thing that the cell membrane does, although we know that it encloses, is its term, it is selectively permeable. Whereas the cell wall will let everything in, this whole structure of the cell membrane allows things to move in and to move out, so we can cross, so we can control what we need to look at and what we need to. So what comes in, what comes out, we can have control over that. Okay guys, I'm afraid that's all we have time for now. We're going to have a quick break and we'll be right back. Welcome back, Life Sciences. I hope you had a good but a quick break. Get that oxygen flowing, get the blood flowing. It clears the mind and helps us to study and remember better. We are recapping the cell today, right? We're looking at the concepts. What is a cell? How does transport occur in the cell? And all of these functions, right? These organelles that we're going to look at that we find in the cell that have a particular function. We ended off last time with looking at the concept of what the cell membrane was. Now, if we have a look again at the structure of the cell membrane controls what is going to move in and what is going to move out. That's a really important concept when we're looking at this whole selectively permeable. I know another word is differentially permeable that you often use, but as I said, I can spell that better, and that's why I use it. Now, the cell membrane is going to control what moves in and out of the cell. Now, when it comes to transport in the cell, things need to move. And as I've said before, how do we get things moving? We're going to speak about a concentration gradient. And what we're talking about, a high concentration means a lot of something. And a low concentration means less. Now, as I said to you before, imagine this is a hill, okay? What would be easier for me? To get things up the hill or to get things down the hill? And the easiest is to go from a high to a low. And your body feels exactly the same way. Why must I spend energy on doing something that's going to be energy expenditure when I can do things the easier way? Remember, it's not so much about the easiest way, but it's what is efficient. So I've got to move things around my body all of the time. Plants have got to move things around their structures all of the time. The easiest we can do it, the less energy we can use, that's going to be far more efficient. So how do I move things in and out, right, of a plant or an animal? The first thing I'm going to look at is diffusion. And simply what diffusion is moving a substance. Here's my high concentration. If I look over here, this is going to be my low concentration. So what the molecules want to do is they're going to go from a high to a low, and they are going to spread equally. Okay, I'm going to show you a concept, right, that you're going to come across later. So if, for example, here I'm going to draw, this is a leaf. Okay, and at the bottom of the leaf, there are little holes that can open and close. They are called stomata. And what happens is a plant needs to photosynthesize, and the plant needs carbon dioxide. So what happens is the plant will always bring carbon dioxide will move from a high concentration out of the plant into a low concentration in the plant. Now, I know this is a weird concept for you, but what has it been able to do? That process of diffusion, moving something from a high to a low, actually enables, right, the plant to photosynthesize. Exactly the same happens to us. In our lungs, all right, when we breathe in, oxygen comes into our body and moves from a high concentration via diffusion, because it's a gas, to a low concentration in our blood. And the carbon dioxide moves from a high in our blood to a low, all right, in our lungs. That's how we breathe. That's how we can bring air in. We can bring in nice fresh air, bring out. So all these parts have it, how we're going to move things around the body. So diffusion is one part. The next concept, all right, is osmosis. Now we follow the same, all right, the same concept where we're going to move water. Right, now this is really important. What moves? Water must move from a high concentration to a low concentration. Now there is a bit of a terms and condition here, because when water moves, it must move through a semi-permeable membrane. If I've got a little jug over here and I put water in here and the particles just move around, that is going to be diffusion. But if you have a look over here, what I need to do when osmosis occurs, okay, is that I need to have, all right, a concentration gradient. Now, if you have a look, this pink is the water. These little blue things are what's dissolved in the water, okay? So say, for example, it could be sugar, it could be salt, and they are dissolved in the water. So I've got more water this side and less water because I've got more dissolved substances. So this has a high water potential. So where does my water move? From an area of high concentration to an area of low concentration. Have a look at the level. The level is going to change, because if my water moves from one area to another, then so is my level of water going to change. Now, guys, this is really important that you understand. When you look at questions like this, and we're going to do one now, you need to look at this. Where is there the most water, and where is there the most dissolved substances? So as I said to you, transport, osmosis, high to low, but through a semi-permeable membrane. Okay, so here is a possible question that we can ask you relating to the process of osmosis. So here we have a grade 10 learners. They set up an apparatus below, right, in the school lab using whisking tubing. Now, you might not have any idea, or you've never heard anything called whisking tubing before, but in brackets, they've explained what it is. They've said that whisking tubing allows water to move in and out like the cell membrane of the cell. So what is it telling you here? It's telling you that this membrane is selectively permeable. I know, as I said, differentially, some of you might even use the word semi-permeable. So let's have a look at the setup over here. So I've got a beaker, all right, and what I've done is I filled the beaker with pure water. So I'm going to change my pen color quickly. This, guys, is filled, all right, with pure water. So I'm going to write here, it has a high water potential. Now what I've done is, if I'm going to change my colors back, I have now put a semi-permeable membrane, this little whisking tubule, And inside here, I have made a strong sugar solution. Okay? So here I've got this concentration gradient. I've got water on the outside, and inside, in this little sac, I've got the strong sugar solution. So what question are they going to ask me? Okay? So they're still carrying on with how they set it up. The learners recorded the level of the water in the beaker before the start of the investigation. Right, so that's telling me that there is the water level is going to important. Next thing they said, after 2 hours, there was a change in the level of water in the beaker. So here is my water level. Guys, 2 things could have happened to it only. The water level could have gone up, or the water level could have gone down. Now, the question is, state the process that is investigated in the diagram below. Now let's have a look. I had water on the outside, I had sugar on the inside, and in between the 2 of them, I had a semi-permeable membrane. So obviously the process that I was going to look at was osmosis. Okay, there we go. I had water, I had sugar, and I had this membrane in between them. Now I have to explain, all right, tell me why the changes that were observed in the level of the strong sugar solution in the whisking tubing and the pure water in the glass beaker. Okay, guys, 2 things they want. Let's break it down. So, what did you notice about the sugar solution, and what did you notice about the water in the glass beaker? Now, osmosis, if we know osmosis occurred, what happened is, guys, where does water move? So let's start. Water moved from an area of high water potential, that means lots of water, in the beaker to, all right, the tubing. So what happened here? Water moved from the beaker into the tubing, which means, let's answer it, all right, the level of the tubing increased. There we answer the second one. Okay, then that's the next thing. So as the pure water moved out of the beaker, the level decreased. Okay, so guys, what happened? This level got lower and this got more. Because where does water move? From a high concentration to a low concentration. Okay, so those are kind of questions that you're going to get. Have a look. Do you have a concentration gradient? Do I have a high water potential? Do I have a really strong concentration of sugar or salt? That is usually the idea that you're going to have when you're going to do an osmosis experiment. So just keep that in mind when you do that. As I said to you before, just make sure when you get these that you're able to explain the process of osmosis. Welcome back, Life Sciences, from your short break. We are revising and we're bringing up all those old things that we look. So the breaks are really important just so that the blood can flow and the oxygen can flow and can make us clearer. We are recapping the cell. We're going over all the important concepts. We've looked at the cell membrane and how things are transported across, specifically osmosis. We have looked at the cytoplasm, and as I said, we have looked at the cell wall in plant cells. And now we're going to carry on looking at what structures can we find inside the cell. Now, when we have a look at the structures inside the cell, they are called organelles, all right, little organs. And very often we get the name confused because when we say an organ, then you say, oh, the mitochondria. No, that is an organ is a muscle, the organ is the stomach, an organ is the the small intestine. An organelle is a structure inside the cell. Now, we find that the first structure, when we look at it, all eukaryotic cells have got the structure called a nucleus. And simply what the nucleus is this is our genetic material. Okay, this is the chromatin network. And in eukaryotic cells, the chromatin network, our genetic information, must be within a structure. When we looked at prokaryotic cells like bacteria, you will find that their DNA, their hereditary information is usually just found within the cell itself and not in a specific structure called a nucleus. The nucleus is a really important part of the cell. If a cell doesn't have a nucleus, chances are it's not going to work because it controls all the activities of the cell. However, there is one cell in our body that doesn't have a nucleus, and that is the red blood cell, right? But it doesn't need the nucleus. It just needs to transport oxygen and carbon dioxide. So we're going to not just bypass that one. So what have we got? We have an organelle, a structure in the cell that performs a specific function. So here, all the activities of the cell. The cell's activities, we give the word metabolism, how fast or how slow a cell does something. Right, what else will it have, the nucleus? Hereditary characteristics. And this is going to be very important, guys, because after the cell, you're going to look at the whole concept of mitosis. And mitosis is where the cell divides. Now, if a cell divides and you want to make, so if I have a cell and I want to divide into two identical cells, what must be identical? Their nucleus, because all the information is in the nucleus of the cell. So whether it's a plant cell, whether it's an animal cell, they're both going to have the nucleus. Then what we look at, we're going to find other organelles inside of the cell. Now remember what I said to you, each of these organelles is a specialized structure. What do I mean by that? Is they each have their own function. Right now, you might find that some cells might not necessarily have all of the different kinds of organelles. It depends on the function of the cell, what it needs. So an organelle, as we said earlier, is going to be, a little structure that has a specific function. So all plant cells and all animal cells have the nucleus. What other organelles are we going to find? Now, in the plant cell and the animal cell, we're going to find this very important organelle called the mitochondrion. Now, if I say to you what all living things need to be able to do, I'm going to do Mrs. Krief again on the board. Right, one of the R's is cellular respiration. And what is cellular respiration, guys? That is our energy. Remember when you looked at biochemistry, right? Carbohydrates, we trap the energy, right? When we eat something, right? Why do we eat? Because whatever we've eaten, the sun's energy has trapped that. So when I eat something and it goes into my body, I need to untrap that energy. I'm like a car, right? And the energy is petrol. If I don't put the petrol in, my body is not going to go. My car's not going to go. So as I said, the mitochondria was a very important organelle that we're going to find. And what it did was this whole concept of being able to produce energy. Now I've got a little question here that we can ask. It's a nice little simple question that we could possibly ask in an exam or a test. It says the diagram below represents a cell found in some complex organisms. Now, as soon as we see the word complex, we know that it is a eukaryote. The enlarged section represents an organelle. Now, guys, this is one of the trickiest things you need to do is very often you're given a diagram and you need to be able to see how the organelle is very often represented. It says the enlarged section represents an organelle labeled X. Now you're going to find very often, if I go back to a mitochondrion, that it looks a bit like a worm. All right, when I draw it, I always draw it looking like that. And that's very similar to what they've done over here. So let's say the enlarged section represents an organelle labeled X. Now you want to know what the X is. The fact that they've given you a few of them is also a hint as to the answer. Okay, the first question is identify it, all right? And as I said to you, very often if it looks like a worm-like structure, it is going to be the mitochondrion. Now, guys, what is the difference between mitochondrion and mitochondria? It's a singular and plural. You can write either. It is not a problem at all. State the process that this organelle performs. Now, you see the problem. If you don't know what the organelle is, then you're going to have a problem with the rest of the question. So the organelle we said was a mitochondria. What is the process? The process is cellular respiration. Okay, so that's a really important concept that we need to understand. Identify one molecule produced by this organelle and explain why, all right, this molecule is important to the organism. Now, we said, let's go back over here, that the process was cellular respiration. But although we haven't learned the process of cellular respiration, we've looked at the mitochondria and we know that what does it need? Have a look over here. I'm going to go two back. It produces ATP and we need it for energy. So if I go to this question, what's the one molecule produced? ATP. And why is it important? We need it for energy for our body to work, to function. So guys, all things in our body, anytime our body does anything to keep warm, to move, when we think, when we grow, when our cells heal, right, we need energy in order to be able to do that. Okay. Then we're looking at the next organelles that we're looking at. Same as the mitochondria, we find these in both a plant and an animal cell. Now these organelles have a very close relationship with each other. Now later on, right, in your studies, you're going to have a whole concept of how proteins are made. Now, proteins are really important, and a lot of the proteins that are made in your body, right, are life-giving proteins like hemoglobin that you need to transport oxygen, or blood clotting proteins, or insulin that you need to help you with your sugar. So if you don't produce it, that could be quite a problem. So what do these three have in common? So generally, if I have a nucleus, the nucleus has these branching tubes that go throughout the cell, and they usually end at the end of the cell. And this, guys, is a transport. All right? It's a transport system for the cell. You'll see that it starts at the nucleus and usually might end at the cell membrane over here. Now, very often, and that is called, okay, this transport is called my endoplasmic reticulum. And I usually can abbreviate it to ER. Now, what else it does is, on the ER are little structures called ribosomes. They are non-membranous structures, and that's why they can be found in bacteria, the prokaryotes. Guys, ribosomes make proteins. Okay? And what happens is these ribosomes are on the endoplasmic reticulum. What can happen is they make the proteins and the endoplasmic reticulum can store them. Now, when we look at the concept of a protein is an extremely convoluted structure of, all right, when we come to look at how it's made. So what happens is when the ribosome makes the protein goes to the Golgi body or Golgi apparatus. And what the Golgi body does, it takes that protein and it packages it. All right, and it packages and it sends it, there we go, sends it out of the body, right, to be used. So these three organelles over here have quite a close relationship with each other. And as I said, we're going to see it in both plant and animal cells. Okay, guys, that's all we have time for now. We're going to take a quick break and then we'll be right back. Welcome back, Life Sciences. I hope you enjoyed your little break. We're going to be recapping the cell today. We're looking at all of the things that the cell has to do. We've looked at this whole concept of the boundaries, a cell membrane, a cell wall, if it's a plant cell. What do we find inside the cell? It's the cytoplasm, as you must remember, this jelly, right? And we're going to find these structures called organelles inside the cell. And that's what we're busy looking at. The last segment that we looked at the endoplasmic reticulum, we looked at the ribosomes, we looked at the Golgi apparatus and the nucleus. And a lot of these things are not going to make too much sense for you now. But as I said to you, this is the foundation. As we go on, all the organelles and the importance that they play in the functions of the cell are going to become much clearer to you. You must just be able to understand the basics. So we're looking at the concept of organelles inside of the cells. And we looked at last time, as I said, the nucleus. We looked at the endoplasmic reticulum, the ribosomes, and the Golgi body. Now, plant and animal cells all have those. Now, some organelles we're going to find are going to be only found in a plant cell or have a much greater function than in a plant cell. And the first one that we're going to look at is these plastids. Now, what on earth is a plastid? A plastid means plast, which means a color. Now, plant cells, very importantly, can photosynthesize. And what do we mean by that is they can make food. So when they make food, they also need to store food. Right, so what can plants do? Now, the first one, as I said to you, the first thing you need to be able to do for plants are able to photosynthesize, right? We as animals, we cannot, we are heterotrophs, we need to eat. Plants can photosynthesize because they have got this very special plastid called a chloroplast. And basically what happens is you'll look at it later on in your life science chapters, that when sunlight hits the chloroplast, a whole lot of things are going to occur. And at the end, what is going to happen is they are going to make the food. Now, what plants do is they are going to store the food. And how they store it is going to be in these cells. And when you do plant tissue, you're going to hear the cell called parenchyma. But if you have a look over here, each one of these is a cell. Now, are you wondering, why is it blue? The reason why it's blue is each of these cells actually is storing starch. All right, so the plant made the food, and now it's going to be stored. And what is a leucoplast? It means something that's colorless. Can you see here? If I didn't have the blue, it would be white. And what it actually is, all right, is storing is starch. Now, why is it this blue color? Because if I put iodine, right, if I stain it with iodine, let's go back to my food tests. What happens to starch? It goes a blue-black. This, guys, would probably be your potato, right? Your potato has got leucoplast. They're your potatoes. They are your storage of, right, those starches. This is a chromoplast. Now, chromo means color. Now, guys, this is probably what your tomatoes look like. And what are tomatoes? Once again, right, we have got food that we store, but we have a pigment, and usually we call it beta-carotene, right? It's a color pigment, but it's a bit of a vitamin that we're going to find in our plants. Another way we're going to find chromoplasts is in our flowers. All right, a flower's got colors, and those is where, that's the chromoplast, is where we're going to find those. All right, guys, the next organelle we're looking at is vacuoles. Now, a vacuole, I've written here in a plant cell, not only in plant cells, we're going to find them in animal cells. Vacuoles in animal cells are a lot smaller, and sometimes they're only temporary. For example, if it eats or if it needs to get rid of a waste product. However, in a plant cell, if you can have a look at the diagram in the board, the vacuole, this is the whole vacuole, almost takes up the whole cell. And what it does is inside here, it stores water, all right, and minerals. But now what happens is that when it stores it, it fills it up and it pushes against the cell wall. And when it does that, we say that the cell is turgid. What do I mean by turgid? So remember, when we look at a plant cell, as I said before, if I cut it open, I'm not going to see a skeleton. So a plant cell is reliant on its cells and its tissue to be strong. So we've got now imagine, we've got a strong cell wall that's rigid. And when a plant is well watered, this vacuole fills, imagine it's like a balloon, and it keeps the cell turgid. Turgid means upright. Now imagine what happens is when I don't water the cell, plasmolysis. So the water goes out from a high concentration to a low. And what does my vacuole do? It shrinks. And what happens to my whole cell? It collapses, it wilts, and I water it, and all of a sudden, there I give my plant shape again. So the vacuole, I won't say it has a more important role to play in the plant than animal, but in the plant cell itself, the vacuole plays a really important role. So as I said, it stores water, and you can see it takes out the whole cell. It pushes, this is the nucleus, and this is the chloroplast, pushes everything to the side to be able to make the space. Okay, guys, the next one we're looking at are called centrioles, and they're very difficult, all right, organelles to try and explain. And they only become clearer once you look at the concept of mitosis. We only find them in animal cells, and there are two of them, and they usually lie at right angles to each other. And what happens is during mitosis, these little guys, if I were to draw something, are going to produce what we call spindle fibers. That's only a concept you'll see later. And what they basically do, imagine they're like fishing rod. They're going to attach to like a chromosome, and they're going to pull, and that's what how they're gonna move things apart. But when you have a look at mitosis, their role is going to be very clear. So they have a role in how the cell divides. And they're only visible in an animal cell. For some reason, we don't see them in a plant cell at all. Okay, guys, so let's have a look at common questions that we can ask on this section. Very often you are given a diagram of a plant and an animal cell, and you need to be able to answer questions, tell the difference, etc. Okay, so the question is, the following diagrams represent different types of cells. Okay, so what do we have to answer? Identify the cells represented by diagrams 1 and 2. So you have to look at it. The first thing always that you need to be able to identify is which one is a plant cell and which one is an animal cell. Okay, so this one is a plant cell, right? And the reason why I know that is, what do I look at? I can see the shape is quite regular, but I have a look. I've got double membranes over here. I can also see that I've probably got chloroplasts, where in the animal cell that is missing. So plant 1 is a plant cell, right? And plant 2 is an animal. All of a sudden I couldn't spell animal. All right. Now it says, give the number and name of the organelle which serves as a storage site for water and mineral salts. Now you're looking for the vacuole, that is the name. And remember, vacuole, usually I'm going to look in the plant cell because that's where it's going to have a far more important function. My answer here is going to be number D, because can you see it? It's a nice large open space. There's actually 2 of them in there. So my number is a D, and the name is going to be a vacuole. The next question, provide the labels for parts A, B, and C. So number A is going to be my cell membrane, because I have one in both my plant and my animal cell. Number B, don't say nuclear membrane, go for the whole organelle. All right, so number B here is going to be my nucleus. And part number C is going to be my cell wall. So guys, you need to be able to recognize all the different organelles, be able to see them. Now the last question before we run out of time says, state whether part D, that's my vacuole, is a permeable, impermeable, or selectively permeable membrane. Guys, it's this, and the Y is because it is surrounded by a membrane. And what do we know in our cell? That all our membranes, right, are going to be selectively permeable. Okay, guys, go and do revision exercises. Go and find past tests if you're able to. Just practice, practice, practice. That's all we're going to have time for in recapping the cell. Until next time, cheerio, bye.